Good afternoon. My name is Brandon Miles, and I'm here to talk about the gut microbiome experiment that we have conducted in class. I would first like to talk about microbiome and what it actually is. It plays a huge role in human health and diseases. The microbiome is a collection of microorganisms that include fungi, bacteria, and viruses living in a given location. The microbiome plays a huge role in sociological conditions such as the IBS or irritable bowel syndrome. And this connects to gut microbiome because we believe that, well, everyone knows that gut microbiome is always at homeostasis, but we believe when gut microbiome is imbalanced or dysbiosis, that can mean that can be the starting points of IBS. Our hypothesis was for the experiment is the difference of gut microbiomes in a guinea pig and a hamster. We had two samples, an 830 and a 620, that were conducted in the experiment. The 620 was a 3-year-old guinea pig, American tricolor guinea pig. His name was Pef. He was about 14 pounds, exercised daily, had a great eating habit. He ate veggie, had a veggie supplement for about 3 years, and he also was able to interact with his brother. 830, our second sample, was a hamster. His name was Prince. Prince's health was average. It was pretty good. He also exercised daily and also interacted with his kind. Right now, I want to talk about the major steps of our experiment. We started off by isolating DNA using the Kagan kit. Then we used Zymo kit to concentrate DNA. Next, we used the UV spectrometer to estimate the purity and concentration of DNA before the PCR, which amplifies the bacteria 16S ribosomal RNA. The first sample we had the first sample ratio was 100 to 117, and the second was 100 to 5.36. We then used gel electrophoresis, which was to check the products of PCR at 250 volts. We then used the Agent Core Ampure XP kit to remove the sequence amplified PCR buffers. PCR indexing, which was the next step to move on. We used two primers. The 3N was attached to the amplicone, and the 5N was attached to a different sequence sequence tags. PCR reactions went through the thermal cycle for about 8 cycles. There was a 72 degrees Celsius, 55 degrees Celsius, and a 95 degrees Celsius that all went through for at least 30 seconds. We had to do another cleanup for the volumetric DNA quantification step, and that's just determine the concentration of amplified PCR cleanup. After the PCR cleanup, we used the Quipit fluorimeter because it is better than the UV spectrometer. The Quipit fluorimeter is able to distinguish RNA, protein, and DNA, but UV spectrophotometer cannot do that. So that's why we use the Quipit. So now I would like to talk about our DNA, how our DNA was shown. So I'm gonna bring this a little closer, and you can see that in lane 1 contains positive samples for bacterial DNA. Lane 2 contains sample 830, and lane 3 contains sample 620. Lane 4 contains the negative controls, and the other lanes are controls and samples from another group. Unfortunately, our samples were accidentally thrown out, and we had to use a random DNA sample. We had to change the samples from 830 to 840 because there was no data for 830, and also we thought 840 was the most similar to 830. We also used 620 from a previous class, which was used in the DNA analysis. Now for our kingdom chart, or kingdom graph, I should say. There has shown that bacteria, as you can see, bacteria in both 620 and 840 was 100%. There was no unclassified percentages. In our order graph, there were 3 out of 6 bacteria that were common. There were Clostridiales, unclassified, and Bacteroideales. The percentages were very different. For example, Bacteroideales only 0.15% of sample 840s read, but 25% of the samples 620s read. In the genus graph, sample 840 was Clostridium, which makes up 59.39% of total reads. And the basic definition of unclassified or other basically means that there is a DNA sample that cannot be identified as aluminum. Okay, so electrophoresis. Electrophoresis is the extraction of DNA, and how successful because of the fluorescent lines. As seen in the pictures indicated, when a concentration is high, there should be more than 100 nanograms per 50 ULs by UV nanodrop spectrometer. Basically, this means that there is enough DNA in the sample to represent the distribution of bacteria in the gut microbiome. In our original samples before they got thrown out, unfortunately, they were higher than 100 nanograms per 50 UL. So that means they contain enough DNA to go through the accurate analysis. PCR reaction was successful with both samples, and their expected values and controls did as well. In the positive controls, they had a 550 base pair mark according to electrophoresis, to let us know if there was a different DNA segments. And the negative control, they did not have any DNA, but showed a faded line that represented a cross contamination, but not anything to be worried about. For both samples, there was a strong. For both samples had strong lines of 450 base pair, which trails the DNA, which is most likely because of the high concentration of DNA in both samples. So that's why it looked like that. But knowing this, we see that the reactions worked by DNA fragments found in positive control match with the samples. And there was no presence of other types of DNA, which meant the bacteria DNA has been successfully isolated. So now I want to compare both animals. And sample 620 had 20,694 reads, and for sample 840 there was 13,423 reads, and both numbers were far above the amount of reads needed, which is about 1,000. And our hypothesis was supported because DNA analysis showed many differences between sample 840, the differences between sample 840, which was a ferret, and sample 620, which was a guinea pig. This can also be seen through the different percentages of similar orders reads and through the orders of two animals that did not share. And lastly, the similar genesis between the two animals, other than unclassified, also shows the different also shows how different the gut microbiomes are.